So first of all, welcome to the Chappel lecture. It is indeed my pleasure to see so many faces here, and I believe that we have more than a dozen or so people joining us online. Last statistic I had was 18 people, so we might even have more by now, but we're just going to move along. I know that we are 5 minutes late or 6 minutes late. As always, we have some sort of technical difficulties, but hopefully this is going to be the end of our technical difficulties. And Dr. Kaushic and I are going to have to be like conjoined twins for the next couple of minutes or so that I can do the introduction, because we don't have too many microphones here. So let me just, first of all, my name is Shayan Sharif. At the moment, I'm the acting dean for the Ontario Veterinary College, and it is my honor and pleasure to present to you this year's Chappel Memorial Lecture. The objective of the series is truly to bring each year to the University of Guelph campus a scientist of international repute to address interested members of the student body and faculty. And I see a lot of our students and a lot of our faculty, retired faculty and faculty who are actively involved in research and teaching. And through such an encounter and through further informal discussions, the university hopes to provide its members with an opportunity to hear people on the forefront in their own research sphere who will not only summarize their current work, but they also trace its development and progression over the years. And the Chappel Memorial Lecture is intended to be not so much a delineation of a field of study, but as a source of inspiration for those engaged in or considering getting into graduate studies in the general field of biomedical research. And it's my pleasure to introduce to you Dr. Charu Kaushic. Dr. Kaushic is no stranger to many of us. We've interacted with Dr. Kaushic over the years, and I know that Dr. Kaushic has been one of the avid supporters of OVC and the concept of One Health. So it's indeed a great pleasure to have Dr. Kaushic here. So Charu is the scientific director of the Canadian Institute of Health Research, Institute of Infection and Immunity, and she has served in that role since 2018. Dr. Kaushic is also a tenured full professor in the Department of Medicine at McMaster University, not too far away from us. In her role as the scientific director for the Institute of Infection and Immunity, she has been responsible for making decisions for CIHR, Canadian Institutes of Health Research, strategic investments in the area of infection and immunity, nationally and internationally. She also represents CIHR and Government of Canada at various national and international forums related to infectious diseases. During the COVID-19 pandemic, she was very closely and intimately involved in shaping CIHR's research response to the pandemic and served on Canada's COVID-19 National Immunity Task Force and Health Canada's Variants of Concern Leadership Group. Dr. Kaushic has a PhD in immunology, and she did her postdoctoral training in mucosal immunology.
Okay, so delighted to be here and delighted to get the technical part out of the way. So this is the program that I was talking about, the CIHR Research Excellence, Diversity and Independence Early Career Transition Award. It's meant to address the gap that we see in Canada's academic pool, which does not match with our population or with the diversity that's seen in most universities in Canada. So it's aligned with the Government of Canada priorities in terms of building a more equitable and diverse faculty pool, and which will also reflect in the research that they do. So that's what it's aimed at, and I'm not a Mac person, so. Nope, I got this. I'm just going to use the clicker to do this, and I'm just not going to touch the mouse. So, okay. So this award it's just outlines some of the details. If you're interested or you want to have apply yourself or have others interested in apply, you're welcome to go to CIHR website, and you should be able to find if you just search up CIHR REDI program. And the deadline is coming up soon, so people should have started already or working on it. It has two different phases. There's a mentored phase, which is for senior trainees with some funding for salary as well as for conducting independent research. And there's a phase 2, which can run to up to 4 or 5 years, which has partnered money that's available, and the partnership is with the institution that's offering the faculty position. So in total, CIHR will provide money for 6 years. This is the first iteration of this, so we're going to see what are the lessons learned from it. It is focused on specific populations. And again, we'll see how that works out. And their goal is that in future to then open it up to more categories of people. Okay, so I want to start out by talking a little bit. Okay, that looks different than what I had on my computer. So is this going to mess with? Okay, there we go. Okay, am I echoing? If I don't move, will it go okay? Okay, so just want to talk a little bit about my lab's research program. So as I said, I've been at McMaster for 23 years now. And my lab does in vitro, ex vivo, in vivo clinical research to improve women's reproductive health. The main focus of the lab is mainly to elucidate biological factors that lead to disproportionate susceptibility to sexually transmitted viral infections in women. And we particularly look at 2 infections, HSV2 and HIV. However, in collaborations with many other groups, we have looked at a whole slew of different sexually transmitted diseases, including bacterial diseases, chlamydia, gonorrhea, candidiasis, which is a fungal infection, as you would know. And then the second one is to determine how we can utilize this understanding to develop prophylactic and therapeutic strategies. So that's where the focus of the lab is, and then different projects will use different approaches, whether it's in vivo mouse models, in vitro models, clinical studies, etc. So what I'm going to talk to you today about is a snapshot of a particular area in the lab that started, I think, in 2012. I wrote a grant to CIHR, a team grant to CIHR, because my lab had done quite a lot of work on looking at sex hormones and how they affect immune responses in the female reproductive tract. And the area of microbiome was very hot and coming through there, and it was clear from early publications that the vaginal microbiota has a significant and profound effect on the outcomes to sexually transmitted infections. And we wanted to get a little bit more in depth into it, and there was very little information on how other factors such as sex hormones will interact with microbiota. So we wanted to get into that. I was successful in getting the team grant, which ran over 3 years. And we started multiple areas of work. So I'm going to summarize some of the clinical studies that we did, how we validated those clinical studies using humanized mouse model. And then based on that, we've now just finished a clinical trial, phase 1 clinical trial. So just a bit of background on these topics, and then I'll get into the data part of it. So I'm not sure how many people are familiar with the HIV statistics in this room. So just a few basic statistics. This is a little bit outdated slide, but the number of people who are living with HIV, it has hovered around 38 million for a long time now. And the reason for that is that even though there are antiretroviral therapies, there's a lot of drugs that are antiviral that are available in different parts of the world, the number of new infections is just not going down, and the number of people dying with infections is also remaining very steady. So that is the reason why the number is in equilibrium, because the number of people who are dying from HIV is approximately the same as the new infections that's happening, around 1.2 million. So the overall numbers keep hovering around 38 million, but underneath that there's a lot going on. You'll see from the slide that the area in red, which is the sub-Saharan Africa, has the largest number of HIV infections, of people living with HIV infections, followed by West Africa, Asia, and then it goes down that list. So in comparison to that, North America has relatively fewer numbers, but also there's a difference in the type of infection or who has HIV infections in these different geographical areas. So on the left side, you will see that there's a very specific type of population that is getting infected with HIV, new HIV infections, and people living with HIV. And if you look on the left side, that outlines a typical what's happening in Southern Africa, where there is the largest number of people living with HIV. In the blue bars, you will see the female prevalence for HIV infection, and in the green bars, you'll see the male. And right away, you can see two things. Number one, very high prevalence rates. You can see that in most countries, and these are from 5 different countries in sub-Saharan Africa, very high prevalence rates, between 15 to 30%. And the second thing you'll notice is that it's much higher in women than it is in men. So there's a gender bias here in terms of the prevalence. If you look on the right side, this is a typical profile of what it looks like in high-income countries. And this data is from the United States, collected up to 2010. And there you will notice, and I'm not sure if the size of the graph is large enough for you to read at the bottom, but the first bar is white MSM, then it's black MSM, and then it's Hispanic Latino MSM. So clearly in high-income countries and in North America, and the Canadian data looks very similar, HIV is still most prevalent in men who have sex with men community. And then if you look at that, you can see black heterosexual women, black heterosexual men, and then white heterosexual women, so it goes down that scale. So again, there's a difference between race and gender, even in North America, on who is who's living with HIV and who has the most infections. In Canada, we have recent data that actually the number of new HIV infections in African, Caribbean, black communities matches that of what's happening in Africa. So that's very worrying for us, especially in young women who are immigrants or are living here. They have very high susceptibility to HIV-1. And the other thing that I want to point out is that when we think about HIV, in an HIV field, we've been doing this since 1980, so it's been about 40, 45 years. We spent a lot of time looking at HIV in blood because it was the easiest thing to do. So a lot of the first developments and the therapies and everything came from what we measured in blood, how much virus is there, how it circulates, what happens to it. But in reality, for prevention, you need to address where HIV starts. And HIV is a sexually transmitted infection. So it's a primarily mucosal infection that starts on mucosal surfaces. And that's what this table actually shows. So on the left side, it's a pretty wordy table. So I just want you to focus on the leftmost side, which is the HIV invasion sites. And if you'll see female genital tract, male genital tract, intestinal tract, placenta, and bloodstream, the main places where the infection starts. And then focus on the rightmost column, which is really the estimated contribution to HIV cases worldwide. And you'll see right away, and for easy math, I put it down at the bottom, about 40% of new HIV infections start in the female reproductive tract. And more than 90% of HIV infections start on mucosal surfaces, whether it's through homosexual transmission, heterosexual transmission. It's primarily sexual, majority of it happening through female reproductive tract, showing that it's more susceptible. The only other mucosa that's more susceptible than female reproductive tract is the rectal mucosa, and therefore MSM have very high risk for acquisition of HIV in unprotected sex. A little bit about the immunology and anatomy of female reproductive tract. Probably this is something familiar to at least some of you, if not all of you. So I'm going to focus primarily on female reproductive tract because that's where my lab focuses the most, because that's where a lot of the infections are started. And the anatomy and immunology of the reproductive tract, we've tried to dissect this out here for you. Basically, the upper reproductive tract, which is the endometrium or uterus, is where the fetal growth occurs. And it has a very different physiology as well as the anatomy than the lower genital tract, which is the vaginal tract. So we've outlined sort of what the vaginal tract and ectocervix looks like. It has a different kind of epithelium. It's a squamous, multi-layered squamous epithelium versus the upper reproductive tract that you can see on the top right, the uterus and endocervix. It's a columnar epithelium, so very different epithelia with very different metabolic functions. And then the cervix, which is shown on the bottom right, you will see that it's unique in the aspect that that's where the transition from the columnar epithelium to the squamous epithelium happens. And at this point is where there's the likelihood of most infection actually occurring. Interestingly, this is also where you find most inflammation. So women that have inflammatory conditions have accumulation of T cells in this area. So you can imagine that HIV loves this area because HIV loves inflammation. It loves having activated T cells sitting just underneath the epithelium. So cervix is a particularly susceptible site for HIV transmission. So let's talk a little bit about vaginal microbiome, and then I'll jump into some of the studies and the data. So vaginal microbiome, I know having talked to some of the students, there's good work happening here in the microbiome area, but maybe not everybody's familiar with the vaginal microbiome. So the vaginal epithelial lining, like most of the mucosal surfaces, is covered with bacteria. Millions of bacteria. However, compared to gut, it's very different because in gut, and for many of you who may be working in the gut area, the more different types of bacteria you have, the better it is. Exactly the opposite in the vaginal bacteria. It's dominated by lactobacillus. So for women to have what I won't say healthy because that's not a good word to use, but optimal bacteria in their vaginal tract, it must be predominantly lactobacillus dominant. So the reason is that some of the species of lactobacillus make all kinds of lactic acid and other things that lowers the pH to around 4, and that creates an inhospitable environment for sexually transmitted viruses to easily come through. So this environment is protective against STIs, and it really doesn't affect the other physiological functions. Like it's not affecting the semen because semen carries an alkaline pH, so it neutralizes. So there's all kinds of different measures that are in place to make sure that reproductive success is not compromised, but there is a barrier to sexually transmitted viruses coming in. On the bottom, you can see that when there is a disruption of this lactobacillus dominant environment, this leads to local inflammatory responses. And it can lead to easy disruption of the barrier function that the squamous epithelium has, and easy transmission of the virus can occur to this. We know this because a clinical condition that's common that's related to the vaginal microbiota called bacterial vaginosis, where there's not a dominance of lactobacillus but other bacteria, and there are certain clinical criteria that go with it. There's AMSL criteria, there's Nugent scoring so based on clinical criteria that correlate with absence of lactobacillus, there's a very high likelihood, 2 to 5-fold increase in HIV infection. I just want to flag the ethnic differences in vaginal microbiota. The reason that this is important is that this is one of the biological factors that we are looking at to see, are these differences in microbiota correlate with increased HIV susceptibility. So a lot of what I'm going to talk about is going to focus around that area. So on the top panel, you will see a typical microbiota for African American women. And you will see on the y-axis on both the graphs are different kind of bacteria. So you will see, all I want you to notice is where the blue ends, because the blue and the yellow are all lactobacillus species. And you'll see that for African American women, and there's numbers in the next slide, it finishes, or the lactobacillus dominant is very small number of women that have that. Majority of the women have that colorful part that's there on the right side. If you compare that to women of European ancestry, you will see that that yellow and blue extends all the way to pretty much the right end of the slide. So majority of the women of European descent actually have lactobacillus dominant, and majority of the women who have black and Hispanic ethnic origin actually have diverse microbiota. And that's one of the things that not just my lab, but a lot of the people are focusing on to see, is this a biological correlate that we can address? And this is just a pie chart showing the same thing that I showed you before, except this with some numbers. And you'll see on the left side, the Asian and white women, and on the right side, the black and Hispanic women. And the blue bar, or the blue part of the pie chart, actually shows what are the polymicrobial or community state type 4. And you'll see that that hovers between 10 and 20% in white and Asian women, but in black and Hispanic women, that number, or CST type 4, goes all the way up to 40% or so. So that is a flag because the polymicrobial microflora correlates with inflammation in the vaginal tract. So I want you to keep that in mind. The other thing to keep in mind is, and this is from a review that was from a little while ago, but it does a really good job of showing the different degrees of species diversity. So the diversity is going from left to right, it's increasing, so more diversity on the right correlates with poor outcomes. And what it shows is that percent of women in each of these clusters, and the more diversity there is, the more there is correlation with different kinds of sexually transmitted infection. And the same is true of negative consequences of reproductive failures and other kind of reproductive issues with the increasing diversity. So that's something to keep in mind. And then finally, the last piece that I want to present to you before we move to the studies is that there is a lot of literature from our group, but also from many other groups, that hormones have a profound effect in the reproductive tract. This is not a surprise. We know that everything in the reproductive tract is regulated by hormones. But the immune system, I'm just going to, hopefully nothing's going to change. So the immune system in the reproductive tract is regulated by hormones. And in general, this summarizes a lot of work done by many different labs. In general, estrogen is protective against HIV and SIV. This data comes from in vitro studies, in vivo studies, a lot from monkey models, SIV models, showing that estrogen in general is protective. Progesterone in general increases susceptibility, but not as much as some of the hormonal contraceptives. In particular, one of a very popular contraceptive called depomedroxyprogesterone acetate, or DMPA, which is widely used in developing countries, particularly in sub-Saharan Africa, has actually been shown to have profound effects on increased susceptibility to HIV. So this has been on the radar for WHO and many other agencies for a while. There's a series of meta-analysis that was done. I like this one because Charlie Morrison did a really good job of the meta-analysis, and they took some very strong evidence, and then you can see with the red rectangle at the bottom that there's about a 40% increase in susceptibility in women who use DMPA, but not other kinds of hormonal contraception. So in general, within the field, if you're using a progestin-only contraception, especially DMPA, there's an increased infection or susceptibility to infection. As soon as you use any kind of contraceptive that has some estrogen in it, for example, oral contraceptive, that risk seems to disappear. But there is an issue with discontinuing the use of DMPA because it's a very easy to use contraceptive, especially in developing countries. It lasts, a single injection of DMPA lasts for about 3 to 4 months. Women don't get pregnant. It's a very discreet form of contraception. They don't have to remember to take pills every day. And in those socioeconomic settings, it's a very popular contraceptive. So there's a whole set of studies that we did before this, but we actually did a study in Kenya where we asked the question of, is DMPA associated with greater diversity of bacterial species in the vaginal microbiome? Because the question was, why is there an increase in HIV susceptibility? Could that be linked back to increased diversity? Because we know that increased diversity leads to more HIV infections. So we designed a study to compare the vaginal microbiota of Kenyan women who are not on hormonal contraceptives with women who are on DMPA or oral contraceptives. And the hypothesis was that the vaginal microbiota of women on DMPA will be more diverse and less likely to have lactobacillus dominance than those who are not on hormonal contraceptive. So the study design, just a high level, it was a prospective clinical study. We'd already done a couple of studies ahead of that because we were working with some of the sex worker cohorts. So we wanted to see what's the baseline of the microbiota in sex workers, and we published a paper before this to show that there's a lot of these are African women who do sex work for a living. So they have multiple reasons for increased diversity. And that was actually shown by the paper that we published for the first time. So we wanted to establish the background on which to do this study, recognizing where we are starting with this. So this was a collaboration with universities of Manitoba and Nairobi. Pumwani clinics, which is the sex worker clinics that are located in Nairobi. That was the cohort that we used, and we had 3 different groups of sex workers, those on DMPA, those on oral contraceptives, and those not on hormonal contraceptives. And because of the high rates of bacterial vaginosis and diversity to start with, it was pretty challenging because we had to rule out women who already have bacterial vaginosis, because how will we figure out whether it's DMPA or just the baseline level is very high. So there was a very strict screening criteria to rule out women who already have bacterial vaginosis. So again, I'm not sure you can see, I just put this here to tell you that even though we couldn't meet our study goals, which was to have 20 women in each arm of the study, or was it 30 women? We started ambitious, 30 women in each study. We didn't think it was such a big deal, but we screened 370 women. And the number of women who had diverse or bacterial vaginosis or other issues, etc., basically after about 9 months of recruiting women, we could only recruit about 22 women in the DMPA and 22 in non-hormonal contraceptive. Interestingly, the group that we had the most challenge with was the oral contraceptive group, and that's just a reflection of what's popular in that population. It's just very hard to find women who want to use oral contraceptives. So I'm just putting this up here. Again, lots of detailed data, basically to show that there really wasn't much difference in the different groups that we looked at, the recruitment, the age, the background, the number of sex partners, etc., look very similar for all the 3 different groups. The only difference was the menstrual cycle stage, because one of the groups was actually on DMPA, the other was on oral contraceptives. Whereas the third group that was not on hormonal contraceptives was obviously cycling, had regular menstrual cycle, and we asked women to come in at a very specific time in the cycle, which was, it's easy to benchmark when they start their periods, and they had to come in to give their samples between 10 to 15 days post the finish of their menstrual cycle. So we made sure of that. One of the things that I want to point out, which is a confounder in the study, but again, it was there throughout, is that vaginal douching is very popular in sub-Saharan Africa, especially in sex workers, because they see it as a cleaning practice. But in fact, it's actually quite the opposite, because douching actually leads to more inflammation. So that's one of the reasons why the rates of HIV are very high in sub-Saharan Africa, because of these hygiene practices that women follow there. Or it could be a contributing factor. So I'm just going to cover a few different areas. So first of all, did we find bacterial diversity was increased? So you'll see on the A, B, C, D, there are just different ways in which we've analyzed the data. And you'll see that by far the one on the top where you see the stars is the increased diversity, KO1, Shannon Diversity Index, all of them. The cleanest graph is on the bottom, right, where you can see that there's a clear difference between DMPA and the no hormone group in terms of the increased diversity. If you put the oral contraceptive, because we had a smaller group, you can actually see on the left side with the circles, and let's see if I can use this. This one, you can see that that actually starts to show a bit of significance, that there may be actually decreased diversity, but we just didn't have enough statistical power to show that for sure. So basically, yes, women on DMPA have increased diversity compared to the no hormone contraceptive group. And then if you look at the what the taxa bar charts show, so each one of these columns is a single woman, and then the color actually shows what kind of bacteria, vaginal bacteria, is present in the vaginal tract of that woman. And what you'll see is that the more color there is, the more different kinds of bacteria there are. And it's divided into no hormone group, oral contraceptive, and then DMPA group. And you can see that both in no hormone contraceptive as well as DMPA, about 68 to 70% of the women actually have much more lactobacillus dominant, but there's a whole 32% or so that have the diverse community. But what's important is to note that little sort of arm of colored colors that are coming out in the DMPA, that tail that's happening all the way to the middle of the group, that's starting to show that there's increased diversity compared to the other groups. The oral contraceptive, like I said, starts to show improvement around 80% of the women, but we didn't have enough statistical power for that. So we wanted to do some other measures rather than just look at the microbiota. And one of the things that we know, the reason for lactobacillus dominance in the vaginal tract is because their vaginal glycogen production under the control of estrogen. So when women start cycling, their estrogen levels go up. Estrogen leads to production of glycogen from the vaginal epithelial cells, and glycogen is a substrate for lactobacillus. So that's one of the reasons why we think that lactobacillus dominance occurs. So we wanted to measure the correlation of whether there's how bacterial diversity correlates with vaginal glycogen. And as you can see on the right, that there is a negative correlation of Shannon Diversity Index and glycogen. So the more diversity there was, the less there was glycogen that was present. We also measured amylase, which is the enzyme that actually converts glycogen into glucose so that the bacteria can use it. And there was a trend towards significance there as well. The other thing that was really interesting is that when we measured vaginal amylase and glycogen between the different groups, you could see clearly that the no hormone, compared to the no hormone group, the women who were on DMPA clearly had lower amounts of glycogen and alpha amylase. So clearly what contraceptive you are on makes a difference to how much vaginal amylase and glycogen that you have. So you can see that on the top between DMPA and no hormones, and then you can see also the same kind of difference between oral contraceptive users and DMPA users on the bottom. Interestingly, we noted that why would there be less glycogen? So clearly there must be, maybe there's some effect on the hormones, and not surprisingly, what we saw was that DMPA has a hypoestrogenic effect. So in the serum levels of estrogen and progesterone were both decreased in the DMPA treated women. So as a side effect of the contraceptive, while it was acting as a contraceptive, it was lowering the endogenous hormones, which is not surprising because a lot of contraceptives actually do that. But because of the decreased estrogen amounts, the glycogen amounts were being lowered as well. The other interesting part that colleagues from University of Manitoba, one of the PhD students who was in Kenya did, was to look at whether this has an effect on the activated T cells of women who are using DMPA. And he looked at many different criteria through flow cytometry, and what they identified was that yes, indeed, CD4 and CD69 and CD4 and CD38, both of which are T cell activation markers, are actually increased in women, and these are non-sex workers. We looked at sex workers and non-sex workers, was increased in the non-sex worker, in the cervix of non-sex worker women. So this was done on vaginal cytobrushes, so this was done locally in Nairobi by Ken Omolo, who actually published this paper a couple years ago. And not only in the cervix, but also in the peripheral blood, he noticed exactly the same trends, that there was increased number of activated T cells. Why is this important? Because it shows that, again, HIV loves activated T cells. If you have activated T cells sitting around in the cervix and in the blood, you can hypothesize that this would lead to more easy infection. So because the study was relatively small, we couldn't get big enough numbers. What we did to validate our results was to actually use a model system. We do this a lot in my lab, where we will use a model and then try to go to clinical studies. But in this case, we actually had a clinical study, and we wanted to go back into a mouse model to see whether we could recapitulate some of these main findings that we had. So we used a humanized mouse model to understand whether we could see similar things happening that we had seen, whether there was DMPA would lead to increased diversity. Now mouse microbiota is very different from human microbiota, but we hypothesized that if the effect of estrogen is same, then we should be able to see mouse diversity, vaginal tract diversity going up as well. So I want to acknowledge my colleague Ali Ashkar, who many of you might know. He is the person at McMaster who originally made these humanized mice. We can get into the model of the mouse if you want. But basically these are immunocompromised NOD-RAG gamma-2 knockout mice that are reconstituted with cord blood. So they basically have, these are immunocompromised, so they don't have most of the immune cells themselves. They are reconstituted with cord blood from infants or at the time of birth. So they reconstitute pretty much the whole spectrum of immune cells within the mouse. And then we use this to actually do some work to see whether we could infect HIV into these mice. So we published a paper before this paper that showed that we could use this model for HIV infection. We showed that it recapitulated many of the features that we see in human clinical infection. So what did we see when we treated these mice with DMPA? On the left, you can see DMPA-treated humanized mice, and on the right, regular cycling mice. And in the gray are the uninfected mice, and in the black are the infected mice. So you can right away see the DMPA-treated mice had much higher rates of infection than regular cycling mice. So clearly, there is this correlation of HIV infection when you DMPA treat mice. But we wanted to see whether this correlates with the diversity that we'd seen in the human populations. So we went back and looked at, and in mice it's easy because you can basically give estrogen or DMPA, so you can do whatever hormone treatment you want. So that's what we did with these mice. And you can see on the top panel that, again, using different types of measures for diversity, the mice that were DMPA treated had much higher diversity than the mice that were treated with estrogen. And then again, we saw that, and that's shown on the left side on the estradiol under the Shannon diversity, that you can see that there's a much lower, as mice are given estrogen and then you measure it on different days, the diversity actually goes down. Whereas with DMPA, it doesn't change. So, and then finally, the piece that we verified was that indeed in these mice, DMPA-treated mice actually also have lower glycogen level. And then we also saw lower estrogen level in these mice as well. The one thing that I want to point out is that in these mice, we looked to see what was going on with the CD4 and CD868 cells, because that was seen to be increased in the human. And again, you can see on the bottom the sum total of what the immunohistochemistry showed, that CD3 cells, so T cells, CD4 cells, which are the target for HIV, and CD68 marker, which is a marker for activation, were all increased in the DMPA-treated mice. So we were able to verify a few key pieces, to basically validate the human study as well. So a series of papers have been published on this. I haven't done justice to the data because there was a lot more work that went into it. But I do want to acknowledge the postdocs and Ken, who was a PhD student, and Julie Lejoie, who is Keith Folk's research associate, and she basically runs the whole clinical setup in Nairobi. And she runs a very tight ship. So we couldn't have done this work if it wasn't for Julie. And then Jocelyn Wessels in my lab, who is actually also another University of Guelph graduate, actually did this fantastic work to kind of pull all these pieces together. So I'm going to switch to the phase 1 clinical trial, but I just want to flag for you that on the left is on the right is what I just showed you. That our hypothesis was that the DMPA leads to decrease in estrogen, decreases glycogen, increases bacterial diversity, increases activation of T cells, and then at least in mouse model, we could see increased HIV susceptibility. What we hypothesized next was that would estrogen do the opposite? Right? If we gave estrogen, would it increase glycogen and alpha amylase? Would it decrease bacterial diversity, decrease activation of T cells and HIV susceptibility? So it's at this point that we switched to doing a clinical trial. So I work a lot with the African, Caribbean, Black communities in Toronto, and we wanted to do this, home-based to see whether we could benefit Canadian women here. So in collaborations with Women's Health and Women's Hands Community Clinic in Toronto, which provides care to about 2,000, 2,500 African, Caribbean, Black, racialized women in Toronto. Many of them are recent immigrants. We wanted to run a study to see whether we could improve the vaginal health of African, Caribbean, Black women. So the study was the estrogen and probiotic treatment of vaginal health. And it's a crowded slide because I didn't want to spend too much space on trying to do this. So what it was a month-long study where women who were screened were found to be qualified. We didn't want to have women who had different kinds of STIs. And basically, they were enrolled in the study. They were given either estrogen ring called E-string, or E-string with probiotic, the Represh probiotic, which has a combination of 2 different kinds of lactobacillus. They were either given it orally or vaginally with the E-string. Or they were just given the vaginal probiotic without any E-string. And we wanted to see whether any of these interventions would improve the vaginal health of women. So the first, of course, this is a phase 1 trial. We had to get approval from Health Canada, so that took about 8-9 months. These are both products that are on the market, but it was off-label use. So we had to go back to Health Canada to get approval to run this trial. So it's a phase 1 trial, so safety, feasibility, tolerability of the interventions was the first goal, but we also wanted to take a look to see whether we were increasing lactobacillus abundance in the vaginal tract or decreasing diversity, both of which would be good. So I'm going to run through these slides a little bit more quickly because basically it's just the phase 1 results that we have put together. So we started this in November of 2019, a phase 1 trial to recruit women in Toronto in 2019. And then of course in February 2020, everything shut down. So talk about bad timing. So we had off and on, open, not open, and then of course the African Caribbean Black community in Toronto was disproportionately affected during COVID-19. So there were not a lot of women willing to come downtown, take public transport, etc. So we kept the study open for almost 2 years. And then we finally pulled the plug on it in December 2021. By that time, even though our goal was to include about 80 women, we could only complete half of that numbers. So I wasn't very hopeful, what we are going to see or what that would look like. So I'm very happy that actually we did fulfill our goal of 80% enrollment and retention to see whether the study or women will even accept this and if this is feasible. So this is just a chart, a flow chart to show how many women were enrolled, how many passed, how many were randomized to the trial, how many received the intervention, and how many completed the study. And basically, 82% of the women who enrolled in the study completed the study. So we met our study goal of about 80%. So that was a relief that if we want to move forward with this, at least women are willing to take this. We also looked at the demographics of the people, and that's summarized in the blue box on the top, that 2/3 of the participants had immigrated to Canada, 88% of in their adulthood. They had regular menstrual cycle, they had very few gynecological symptoms, and about 20% of the women had had one episode of bacterial vaginosis. But if we saw women when they enrolled that they had bacterial vaginosis, they were actually treated with antibiotics and asked to come back to enroll again. So we didn't actually have any women who had bacterial vaginosis to start with. And on the right side, again, it shows the demographics, which were very similar in the 4 different groups. There was not a lot of differences in the different groups, which could account for any differences that we saw. So the first things first, safety, feasibility, tolerability. Here, there is the adherence, which would indicate whether women accepted this. And we had very high levels of adherence. So more than 90% of the women adhered to it, which was surprising to me because we were asking women to insert vaginal probiotic tablets twice a day, and they kept diaries of it. We asked them to record when you would take it out. Did you take out the ring? Did you not use that? So there was very high acceptability. So clearly a very motivated group of women here who were able to do this. So more than 90% acceptability and adherence. There were no severe side effects to these. We didn't expect it, but we needed to do that for Health Canada documentation. So more than 43% of the women did not experience anything. So about 50% of the women, most were very mild, vaginal itching or probiotics, so a bit of gut cramp kind of thing. There were only 3 or 7 episodes of severe outcomes, which were abdominal cramps, headaches, continuous headaches for a few days. But all of those adverse events resolved over the time. And then we did blood studies to make sure in serum there were no changes of the normal, because you're giving hormones. You were required by Health Canada to see if there's any changes in any of the outcomes. There was nothing that was noticed. Glucose levels rose up and met significance, but it was still within the healthy range, so there was no big change. Okay, so let's talk about vaginal microbiota. What happened to vaginal microbiota? So this is the first time I'm actually presenting this data. I want to acknowledge Bibin Gill on the top left, a relatively new postdoc in the lab. She has a metabolomics and bioinformatics background. I was talking to some of the students and staff today that bioinformatics is something that just is something we all have to learn, because we can't do much work in the future without bioinformatics. So that's just the way to go, and that would be a strong recommendation. And at CIHR, we are rolling out a lot of bioinformatics programs to make sure. So bioinformatics today is what flow cytometry used to be 20 years ago. Like everybody had to know if you'd want to do immunology. So the 2 important things, for measuring what would be a positive outcome, I just want to flag here. The diversity, so high diversity is not good, and prevalence of lactobacillus. The 2 don't always go together, because you can have decreased diversity, but you can have some other bacteria that's an anaerobic bacteria that's dominating. So more lactobacillus and less diversity, those are the 2 criteria of what would be an optimal or improved vaginal health. So here's a first look at this. It goes from E-string to E-string and vaginal probiotic, and then I guess I was shifting things around. So it's the 4 different groups. Yeah, the second one is vaginal probiotic, third one is oral probiotic, and the last one is just vaginal probiotic. And the top is E-string. So we looked at, the 3 or 4 different time points. Women were allowed to come back for a 5th visit, optional, just to see after finishing that 1 month, did it continue and did their improvement continue or not. So we looked at it, and the numbers were too low. There was not really much we could make out of it because we started with it was a blinded study, randomized. So women started with different ones and continued. We could see some that had improved, others that had not. So it was very difficult to make sense of the data by just categorizing them in different groups of interventions. I had always worried about the fact that we had too few numbers to see very specific things. So Bibin, who's obviously bioinformatics, so very clever at how best to use numbers to be able to do that. She basically looked at to see how best we could do this. And what she did was different visits in those columns and different CSTs. So 1 and 3 are, 1 is the best that you can have, lactobacillus dominant, 3 is intermediate. Depending on the kind of lactobacillus, it's an intermediate kind of. And then 4 is the one that has most diversity and correlates with poor outcomes. So she tried to plot it out to see how they changed with the visits. Again, very confusing. So we decided to take a different approach and to basically look at changes either in decreased microbial diversity or increased lactobacillus, regardless of what intervention was used. So we had 3 different criteria: a shift in community state type, going from 4, which is diverse, to 1, which is all lactobacillus dominant. Anything in that direction, would be a good change. Increase in total lactobacillus species would be a good change, and a reduction in microbial diversity would be a good change. And then she redid the analysis to see what that looked like. And now we could start to see some trends here. And you can see in that pie chart that about 45% of the women actually had a positive shift using these 3 criteria, but not all together. And this is the breakdown of that group. So the first one is positive CST shift. So of the 41 women, about 19 had overall positive shifts by any criteria. And if you break it down into what category they fall in, so what we would ideally like, they went from most diverse to sort of great lactobacillus dominant. Only 5 of the women had that. But if you start to dig underneath it and say how many women actually had improvement in their lactobacillus abundance, you can see more numbers there. So that adds another 7. Then if you look at women who had lower microbial diversity, you can add another 3 in there. And then if you just look at species shift to a better type of species, there's another 4 women there. So that brings the total to about 19 women, which is about 45% of the numbers. Vibin has a lot of data. She likes to analyze data. We're still chewing over it, so I don't want to get into the details of it. But she had like 30 slides that she sent me 2 days ago. So I shifted and kind of said, okay, maybe 4 slides out of this to start with. But here's kind of like the best summary slide for me is that if you look at the pie chart at the bottom, because by now you should be used to seeing the pie chart, the gray outside circle shows you the lactobacillus. And it went from starting point of about 45% of the women having lactobacillus to visit 2, 3, where about 75% of the women had lactobacillus enrichment. And then even after that, because women came back a week later, some women, a subset of women, many of those women, even though there was no continuation of the intervention, actually continued to have 60, so about 67% of the women had still good microbiota in their vaginal tract. And then if you correlate on the top, you can see that there's a negative correlation to 3 of the bacterial species when lactobacillus increases, which is in the green on the top. There's a negative correlation with 3 species that are associated with BD. And then the bar chart shows the same thing, that there's an increase in the bacteria that are associated with good outcomes, and there's a decrease in bacteria that are associated with negative outcomes. So what does this mean clinically? We showed this to the team, and my clinical colleagues said, but what does that mean for BV treatment? Was there an improvement in BV treatment or not? So that's what I want to show you as the bottom line, that if you look at BV as Nugent score or molecular methods are if you actually do the whole sequencing, you can see much more in detail. And by both of those, there was about 50 to 55% improvement in the bacterial vaginosis status of the women who had those by either Nugent score or by molecular status. The ones that didn't, were majority of the women were already in CST1, which is lactobacillus dominant. So in conclusion, we were able to identify a subset of participants that demonstrated a positive microbial shift by 3 different types of categories that we used for analysis. And about 55% of the women who start with molecular or clinical bacterial vaginosis had a shift in their baseline. The negative shifts were minimal, and those that had, like they reversed, they went from 1 to 2 or 3, shifted back to the original by the end of the treatment. And we could not make any conclusions based on the different interventions we had used. The numbers were too low, and we started with different baseline levels. So there's ongoing work that's being completed. So there's still crunching the numbers and kind of doing a more deep dive into it. As I was putting these slides together, we were still kind of going back and forth and saying, how does this match with this? What's the percentage of this? So we're still doing that kind of stuff. We want to see immune mediators. As my clinical colleagues put it, the proof's in the pudding. Like if you can see decreased inflammation, that should convince us that this is good. So we are now moving to do that kind of work. And then Bibin wants to do, obviously, metabolomics because that's her area. So she's very interested to see what's changing that's leading to these changes in bacteria and what does that correlate with. And I'll finish with an acknowledgement of all the people in my lab, the funding agencies, and especially the women who volunteered to come and do all these studies, both in Kenya and in Women's Health and Women's Hand. And a big shout out to Bibin and my clinical partner, Dr. Fiona Smale, that some of you may be familiar with. So I'll stop there. Apologies for the delays, and thank you for sticking around to hear this.
Thank you so much, Dr. Kaushic. That was fantastic. And also, on behalf of OVC, I wanted to apologize for the delay in getting things started. We have a tendency to say next time we are going to do better, but it seems to be a recurring theme. Every time we are trying to set something up here, as a hybrid method, there is a glitch. So hopefully, we are going to do better. But I know that we are running out of time, badly out of time. But we do actually have some comments here, Dr. Kaushic. Well worth the wait by Emma Berkowski. So thank you, Emma. Having said that, maybe we have time, for one question from the audience, or perhaps, I should give some time to our online participants. For the sake of discussion, let's give time to our online participants because, as an FYI, there's going to be a reception in the OBC cafeteria, and for the people here in the auditorium, please join us for the reception. And I know that Dr. Kaushic would be happy to entertain any questions that you might have. But for the online audience, is there any questions? And I'm monitoring my teams. I'm not quite sure if they can hear me. Hopefully they can. Okay. Okay, so there is a question. The question is, how much bioinformatic experience does your student have? This was very impressive presentation, and some more.
Okay, well worth the wait for sure. Okay, thank you. So Bibin actually has a PhD in metabolomics. So she did her PhD actually at McMaster in the chemistry department, where she basically looked at the environmental toxins in large clinical trials to see the effect of environmental toxins on some of the blood parameters. So she's done serum, urine, blood, bioinformatics, and that's kind of like her field. And I've been wanting to do this for a long time, ever since we started microbiome work, because for me the question is, the changes in microbiome have to come from either the bacteria or the host or their interactions. So we really need to understand what's in that environment, so microbiota versus microbiome. What's happening in that environment. So what are the factors, host factors or bacterial factors? So I've been wanting to do that for a long time, and Bibin, who's a postdoc in the lab, so she has a PhD, is able to bring that area, and she's very independent, which is how the postdocs should be. And so she really, all credit to her for all the different analysis. I just kept saying, but this doesn't make sense, this doesn't make sense. That's our job, right, as PIs, to say, no, this is not making sense to me, Bibin. Is there a way that we could do this? And she was like, okay, let me go back and think about this and do this. So all credit to her for that.
And perhaps just one last quick question from Dr. Diego Gomez. Is there correlation between diet and vaginal microbiota?
Yes, that's very simple. Just like the gut microbiota, the vaginal microbiota. So the very fact that you can pop in oral probiotics and you can see changes in the vaginal microbiota. Right, so Activia yogurt, thumbs up. I'm not marketing Activia, but I'm just saying any kind of live culture yogurt is a good thing to have, probiotics, but of course fermented food and other things that enhance bacterial, yeah. So those are all good things.
Fantastic. In the interest of time, I just wanted to again say thank you. It's been a pleasure having you and hosting you here in Guelph, and we are so honored to have you here.
Thank you very much.
Please join me to thank Dr. Kaushic.